And I would like to thank Dr. Sun-Ki-jan, who is my long-term friend, to invite me for this 20 years ceremony for the IPK establishment. So today I'm going to talk about response and lessons learned during COVID-19 pandemic. Although I have been working for the hepatitis C virus research, but after the Charlie's talk, I choose the COVID-19 for my topic. No disclosure for COI. And just before my presentation, I would like to introduce my institute, National Institute for the Infectious Diseases. But after the experience and lessons, the government, Japanese government, decided to merge the two institutes. One is our institute, NIID, National Institute for Infectious Diseases, and another one is National Center for Global Health and Medicine. To be combined, and new organization will start since April 2025 next year. The name of the new institute will be Japan Institute for Health Security, JIHS. We call JIS, is the new name. And our institute is working on the emergency preparedness and response and the infectious disease intelligence, such as the surveillance and epidemiology research, and also laboratory networks and research and development. So we are very much focused on the research and development for the antivirus or vaccine development. So we probably have the collaboration of the with IPK. And also NCGM has the clinic for the international infectious diseases, and also they have high containment ward, medical ward. So we are focused on the infectious disease control in Japan. So we have the national network for the infectious disease, including local research institute, more than 85, and with the local health centers and hospitals and quarantine stations under the umbrella of Ministry of Health. And also we have an international network, including the Pasteur Institute in France, and also in the South Korea. We have a close relationship with KDCA. We have annual meeting with KDCA and China CDC. And also we have the bilateral, biannual meeting with the Korea FDA and also Chinese FDA to the for the quality control of vaccine and biologicals, such as blood products. So maybe we will start the collaboration and the annual meeting with the KVRI, is a Korean Viral Research Institute, from this August, and I hope we have started the close collaboration with Pasteur Institute Korea. So I will talk about the initial measures against COVID-19, including the laboratory response and also public health countermeasures for COVID-19. And in the second part of my talk, I will talk about the research and development for the antiviral drugs against SARS-CoV-2. So this is to start with initial response for the COVID-19 pandemic. So we had a very happy year end of 2019 and happy New Year on 2020, but on the January 2nd, we had news from the on the ProMed. There is the pneumonia patients hospitalized in the China, Wuhan, and they are related to the seafood market. And after that, we have many news from the China and WHO, but on the actually January 8th, WHO confirmed that Chinese authorities believe that the new coronavirus is related to this pneumonia outbreak. But so, and then January 10th, the genome sequence of SARS-CoV-2 were published, and we started to establish the PCR testing system, and within the 4 days, we prepared the testing system and started the testing for the COVID-19. And the conventional PCR and also real-time PCR method was established, and we distributed them to the local research institute and prepared for the outbreak. And just started the January 14th, we started the testing, and we received the samples on the same day, and we found it is positive on the January 15th. And Ministry of Health reported that our first cases was found on the January 16th. He was a male patient, 30s, and living in Kanagawa Prefecture in Japan, but with a history of travel to Wuhan. And after that, we had the isolated SARS-CoV virus using the cell line is highly sensitive for the coronavirus, because during the SARS virus outbreak, we established the cell line using the Vero cells transfected with TMPRSS2. And the TMPRSS2 expressing Vero cells is highly sensitive for the SARS virus isolation. So we isolated and published announced that January 31, we isolated SARS-CoV-2, and we also published them in PNAS. And this virus strain and its nucleic acid was distributed for the domestically and also overseas within the year, more than 100 cases. And also we need to prepare the rapid antigen test. It is very easy to use bedside or clinic, and it makes a very quick results. But however, we need specific antibodies for SARS-CoV-2. So we try to apply the SARS virus binding antibodies, and very quickly we can develop the first rapid antigen test, and it was approved on May 13th, and the second one was August. And also we collaborated with a company and developed a quantitative antigen test. And it is commonly used for the HCV or HBV antigen testing for the Lumipulse system by Fuji Revio company. And it is sensitivity is small less than the real-time PCR, but it is very convenient, and it takes only 30 minutes for the result. So it is used for the border control measures. And also we developed the whole genome sequence method by the 2 tubes of 49 multiplex PCR, and it was very convenient and useful for the countermeasures for the SARS-CoV-2. This is a laboratory work for the COVID-19, and I'd like to talk about public health response during the pandemic. And I was appointed the chair of expert scientific group for the COVID-19, and I made a weekly media conference for more than 3 years, every week. And the scientific expert group was have a sense of crisis, and we need to provide the scientific advices to the government. And every country has some confliction or trouble between the scientific group and the government. And there is a principle for the scientific advisory group during the pandemic. So experts' role in the pandemic is very simple: provide scientific advice to the government regarding the countermeasures. However, sometimes the information or evidence was very limited, but still experts should provide a certain opinion or recommendation. And also government's role in the pandemic is also very simple. They should decide whether accept or reject for the request or proposal from the experts, because of not only the scientific evidence, but also the government should think about social economy or education and the people's trust, and they all need to consider. So they should decide, but they should explain the reason for the decision, why they are not take the recommendation or take the recommendation. But also governments should also make the decisions amid the various uncertainties. So during the pandemic, it was very important to build a transparent scientific advisory system and gain the people's understanding and trust for their cooperation. And also to make a scientific evidence, that it is very important to have the first few hundred studies And what is the first few hundred studies? This is the clinical and epidemiological surveys to quickly collect the knowledge from the few hundred cases, first few hundred cases. And why first few hundred? When the unknown pathogen emerges, prompt public health responses, such as risk assessment and countermeasures, are necessary. And what should be done for by the FF100 study? So rapid risk assessment, and also provide essential information for the prevention and control of the infection. And the reproduction number, becoming the term of reproduction number, is becoming very famous, popular during the pandemic. This is the indicator of one person infected to how many people on average. So if this number, R0, is 1, the infection is remain a steady state. But if it becomes less than 1, the infection will disappear and extinct without epidemic. But if it is more than 1, epidemic will expand. This is very clear. But there is a very big question and mystery in the early stage of epidemic of COVID-19. We had 9 cases of infected patient were found. And we have thoroughly investigated close contact of the 9 patients. And we found more than 100 close contact, but no one infected. So the no close contact have been infected, so it should be R0 is 0. But it is, the epidemic is expanding. Why it is contagious? The only possible reason is a simple math. Most people do not infect anyone, but some patients infect many others. So in that case, we can explain why the epidemic occurs. So from this hypothesis, the cluster, we call this infection is cluster infection. And from this hypothesis, the cluster countermeasures team was started in Japan. And for the first few hundred studies, we analyzed the first 110 cases, and the X bar shows the number of secondary cases, and y-axis showing the primary cases, and showing the more than 80% of the primary cases has no secondary infection. But only limited number of the primary cases infect to secondary infection. And some of them infect 4, 9, or 11. So infected persons were, and also the infected persons were in the closed and poorly ventilated environment and produced many secondary infections. This is the cluster infection. So if we found that if we can avoid this closed and poorly ventilated environment, we can avoid this cluster infection. So this is a characteristic of COVID-19 transmission mode. And in the left-hand side, it is a full case. This is a homogeneous infection. 1 person infected 2 persons, and then 4 persons. This is R is 2. But in the COVID-19 cases, 1 person infected, in this case, 5 persons. But among the 5 persons, only 1 person makes a second infections. So SARS-CoV-2 infection is highly heterogeneous, and COVID-19 spreads through the chain of clusters. And also, how we can contain the SARS-CoV-2. In the SARS case, the most SARS cases cause severe pneumonia. And also, they became infectious just after the onset of symptoms. So in the SARS case, they can be contained by diagnosis and isolation of those who develop symptoms. But how about COVID-19? In COVID-19 case, they have a wide range of conditions from asymptomatic to severe cases, even death. So and also infection occur even before onset of the symptoms or from the asymptomatic cases. So it is very difficult to contain COVID-19 by diagnosis and isolation of infected individuals. However, as the infection patient increases, the severe ill patient or even death also increases. So how should we respond? So the prediction of at the early phase of COVID-19, the left hand is new infection cases, and also right hand side the severe patient cases. So without any restrictions, the new infections emerges very rapidly, and also the severe cases also emerges very rapidly. And right hand side there is a red line. It is the available ventilator numbers in Japan at that time. So, so if we can freely increase the new infections, the severe cases they cannot receive the ventilator therapy, so it makes very difficult situation. So it is very difficult to contain the virus, but we need to suppress the number of infected people as much as possible. That's a conclusion. So the pandemic response strategy is mainly two theories. The one is containment, slowly contain the virus to reduce the infectious patient to zero. But it makes a very burden for the socio-economical burdens. But medical burden is not so big. And another one is mitigation theory. They accept increase of infected patients and focus on the severe cases care. So in that case, there is a very small socio-economical burden, but for the hospitals, very much burden. And each strategy has its pros and cons, but in Japan, it's in between, between the containment mitigation. We took the suppression theory to suppress the number of infectious patients to reduce the death cases. And we focus on the suppression strategy. But after Omicron emerged, so many countries adopted mitigation strategy because of the fatality or severity is very much reduced. So appropriate countermeasures to control COVID-19 spread is what is the appropriate measures. So Director General of WHO told us the test. So test and isolation of positive patient. But as I mentioned for the SARS-CoV-2, it is not so efficient. And also contact tracing, identification, and isolation, and also cluster measures. These are done by the restriction of infected patients and their contacts. And also it was done in Japan by the infectious control law. And this is a test and isolation PCR positive patient theory. And the course of infected patient, the PCR positivity lasts for 3 weeks. But most infectivity was only during the among the onset of illness, about -2 days of, to the +2 days during the onset of illness. And after that, after the 10 days of after the onset of illness, PCR positive, but there is no infectivity. So PCR positive, but not infectious period. So the PCR testing and isolation of positive patient is not so effective for preventing infection spread. And also, this is a contact tracing. The primary case is there, and infection, and the incubation period of about 5 days, and illness onset, and then diagnosis. So, and also this is a secondary infection about the onset of illness, they infect, and they also had the diagnosis over there. And this is a third infection. So the first primary case was diagnosed and the contact tracing, and they found the secondary infection. But it is very difficult to prevent of secondary infection by test and isolation, contact tracing. And only can possible to prevent third infections. And preventive action to overcome, but still it is necessary, but it is not effective. And also, as I mentioned, the 80% of primary cases do not infect the secondary cases. And also, for the Omicron cases, the incubation period becoming 3 days and the generation time also becoming 3 days. So it is very difficult to prevent even third infection. They only prevent fourth infection. So for Omicron virus, it is very difficult to prevent by the contact tracing. And also this is a slide I already shown that cluster measures is necessary and cluster occurs in the closed and poorly ventilated environment. So where is the infection source? So the forward contact tracing is important to find out secondary infection, but we focus on the retrospective contact investigation. So if we found the infected person, we investigate the before the infection, before the onset, the 2 weeks of the movement, we investigate it and find out the place of the infection. So, and it is very important to avoid places where cluster can form. This is we call the 3 Cs: the closed spaces and crowded places and close contact settings. So this is we highly announced about this and to avoid the cluster infection places. But finally, it is very difficult to control these the infection spread by this method. But finally, we declare the state of emergency and the restriction of general public society and by influenza special measures law for the non-infected population. And the pandemic started from the Wuhan imported cases, and then in beginning of March, there was a spring vacation, and many people going to the Europe and the US for the travel, and going back to Japan and bring the COVID-19 with them, and after that, very much spread the infection, and we declared a state of emergency for the more than 1 month, and the epidemic is slowed down. But still, infection remains and resurgence of European origin. This was investigated by the genomic analysis. But still, we repeated the state of emergency and cluster measures, and finally, we had the very small numbers of deaths per year and country compared to European and US countries, and Korea and Japan is relatively done very well. So in the summary, rapid COVID-19 spread like to read medical strain, and the infection mode was droplet infection, aerosol infection, and contact infection. And transmission through the cluster infection and the super spreading event is depending on the environment, so important to avoid 3 Cs. But this infection is very prone to extinction. Effective cluster countermeasures can prevent the spread. However, infection transmission is more likely to continue because there is a difference of generation. Younger generation have mild symptoms or even asymptomatic and have many places for infection transmission. They are likely to gather together, and there is a nightlife and so on. And so we also run the needs medical countermeasures, vaccines, and antivirals. So next, if we have a time, I'd like to talk about the drug discovery research for the COVID-19. This was done by Dr. Koichi Watashi, probably well known about that. And we screened the viral infection experiment. We just infect the virus and wash out and waiting for the 48 hours, and cells are going to die. But if we took the drugs, and if it was effective, the cell survive. So it is very simple assay. And we screened approximately 300 approved compounds. We finished March 2020 by the drug repurposing, repositioning assay. And we found 5 hit compounds. And we found 5 hit compounds: lopinavir, rapamycin, lotenaprovir, and nelfinavir, and cefarantin. And it was effective. The red color shows in the viral infected cells, and lopinavir and chloroquine and the nerfenavir cefarantin can clearly inhibit the infection. And among them, the nerfenavir and cefarantin was strong IC50 and IC90. 90. So we focused on these two drugs. And to analyze the mechanism of action, we did the time of addition analysis, and we found cefarantin inhibits cell entry, and nerfenavir inhibits viral replication. Because of the nerfenavir is HIV protease inhibitor, actually nerfenavir can inhibit SARS-CoV-2 protease activity. So we found these compounds by the in vitro screening, but it really works in the clinic. So we did the simulation prediction. And because of these are already approved drugs, so there is the pharmacokinetics and pharmacodynamics data, and antiviral activity was analyzed, and we simulated that the cumulative viral load can be reduced, and also days can be reduced until the virus elimination by nerfenavir only and also combination. But for the chronic viral infection, there is no different effectiveness depending on when treatment is started. And we know very much antiviral treatment is highly effective for hepatitis C. But in the COVID-19 cases, drug effectiveness is depending on the timing of the treatment. Because of the natural course, the virus dynamics is going down like this. So we need to start as early as possible for the antivirals. If it is late, it is not, we can see the effectiveness. But also the host is different. Somebody is very rapidly decay or very slowly decays. So it is some differences. So we all simulated, although drug show efficacy, different therapeutic effects have been reported in the different clinical trials. But we simulated nerfenavir has a highly activity against SARS-CoV-2. So we planned physician-initiated clinical trials. And that in vitro data was published in the April 14, 2020. And then we planned the investigator-initiated clinical trials with nerfenavir. We chose nerfenavir, and this is the approved drug for the anti-HIV drug in 1889. And we discussed with Dr. Shigeru Kono, the Nagasaki University, and the first patient was registered in Nagasaki University in January 23rd. And we made the multi-center networks in the 11 clinical facilities were chosen in Japan. And this is a trial design, and the control group 60 and drug group 60, and drug was given for 2 weeks, and observation period of 2 weeks. And the primary endpoint was due to virus negative status, and the secondary endpoint was efficacy evaluation, prevent aggravation, and safety evaluation. And the patients were chosen asymptomatic or mild cases. And patients was registered very early, but was stopped because of the drug stock expires at the end of August, and need to import drugs from overseas, and new registration suspended due to delay. And during this delay, the pandemic goes very much surge, and the hospital was very busy, becoming very busy. So we chose the mild and asymptomatic cases, but general hospital clinic, those cases usually visit the general hospital or clinics, but those cases, it is very difficult for the clinical trials. And university hospitals were concentrated, moderate, and severe cases. So in a situation of medical stress, there is no room for the clinical trials. So it was also the difficulties. So we have several difficulties for the operation, facility selection, and infection control, and effort, and securing funds and drugs, and also configuration endpoints, disease characterization, and change in the pathological conditions of SARS-CoV-2. And this is a lesson for the next pandemic, maybe. But finally, we have achieved the target numbers of patients registered in the August of the next year, 2021. And this is a very good effort of the joint team of Nagasaki University and Chiba University. And we made a lot of newsletters. And finally, we published the result of these clinical trials. And this is the patient background, and this is the result. Unfortunately, we did not see any difference, a treatment group and control group. This is days from the registration for the PCR negativity. So we also focused on the patients treated within the 3 days of symptom onset, but still we don't see any differences. So adverse event was not so it is predicted. So this is a summary. The target number of cases were achieved by 14 months. So no antiviral effect or clinical efficacy of nerofilamivir was observed against asymptomatic and mild cases. The safety was for asymptomatic and mild cases was acceptable, mainly due to no adverse events. So but no difference in efficacy against mutant strain was observed. So difficulty, problems, and challenges in conducting clinical trials targeting asymptomatic and mild COVID-19 patients have been clarified. Okay, that's all. This is my presentation. In the first part, I told about initial measures against COVID-19, and second part, our trial for the antiviral drug development. And I would like to acknowledge our team in NIID and other partners in Japan. Thank you very much.
Thank you very much for the one.